All right, welcome everyone. Okay, it's a beautiful, sunny, gorgeous Tuesday, and I know that you all love to be outside. So much more than you love to be in the classroom. Although realistically, you'd probably rather be home in your bed sleeping or in the library studying on Thursday. Nonetheless, we have to carry on and continue with regulation of metabolism. So we've learned about glycolysis, we've learned about gluconeogenesis. Today we're going to finish up on understanding how we can prevent those two pathways from running at the same time and all the different levels of regulation that our body has evolved to make sure we have what we need at the right point in time. You can think of this, so the last lecture, the second class or the last day of class, our penultimate day of class, we will be thinking about metabolism as a whole. So connected to glycolysis, the acid cycle, and electron transport chain, and also thinking about how fats are burned for fuel. We're going to be tying those all together in the last day of class. Today, you can kind of think as a preview or practice round for that last class, but just focusing on glycolysis. You can think of it kind of like a practice round. So really quick, I had a question before class that I wanted to bring everyone's attention to. At some point during the lecture when we were talking about nucleic acids, the convention for the so I just really quick wanted to bring everyone back up to speed on that, and that the alpha phosphate that is closest to the nucleotide. So if you're thinking about ATP, the alpha is close to the A. So that can help you remember and keep the convention straight. So as we go alpha, beta, gamma, the further that we get away from the adenine. Okay. So I just wanted to really quick touch base on that again. You are not responsible for the content from the paper discussion for the exam. However, if there was a question where you were asked to talk about a G protein coupled receptor, you could talk about the mu opioid receptor if you really felt passionate about it and you understood it really well, but it's not something that you will expected to answer a question about for the exam. Really quick, some announcements. So we're going to attempt to do a little bit of group work today, time permitting. In this case, I'll have the instructor be the youngest at the table. Again, the exam on Thursday, but you already know that. I don't need to tell you that. We'll be moving into the citric acid cycle and fatty acid metabolism next week. I'm not planning for there to be pre-assignments for these classes, although I probably will email and suggest that you read over certain parts of the pertinent chapters of the book. And then finally on the horizon, again for that last day of class, we have the final dope-sick reflection and the extra credit assignment. And as we get closer, we'll touch base. I say, let's get through exam 3, and then we can talk about the final. So before we jump back into metabolism, does anybody have any questions that they would like me to answer? Anybody have any other relevant and pertinent questions? Okay, and one more thing, again, we have our review session tomorrow from 6:30 to 8 in Salem 013. So I will be there to answer questions. I will also be proctoring tomorrow from 2:30 for some people who had conflicts with the exam on Thursday. So you're also welcome to stop by Salem 013 during that time and ask questions. I know I have one person coming already, so that can also be considered like an open office hours time where you can pop in if you so desire. Okay, so I wanted to take a quick minute before we dive back into where we left off, and I wanted to talk a little bit more about isozymes and also about the regulation of two isozymes that we spent a good bit of time talking about, and those are the two different hexokinases. So what is an isozyme? Specifically, an isozymes are enzymes that have different structures. So they come from separate genes that have a different DNA sequence, which leads to a different structure, but they do the same job. Now, these proteins could, in theory, be structurally related, but they don't have to. That's not a requirement, but they have different DNA sequences. And oftentimes, this has to do with differences in the environment that they work in. So, some examples of of isozymes that we've seen in metabolism up to this point are malate dehydrogenase and PEP carboxykinase. So we talked about how the first two steps of gluconeogenesis, that are the reverse of the last step, the pyruvate kinase step of glycolysis, bounce between the cytosol and the mitochondria. So we have two different isozymes for some of the enzymes that are involved in that step, in that circumvention process. Lactate dehydrogenase, which is involved in the last step of glycolysis, if glycolysis is your end goal. So turning pyruvate into lactate. There are heart and muscle isozymes. And you may be wondering, why do we have isozymes? Why not just have one enzyme? I keep talking about how lazy nature is, so why not just take one enzyme? It's because not all tissues or even parts of the cell are created equally, so they're going to have a different environment. So for instance, in the case of malate dehydrogenase and PEP carboxykinase, for that step, we're really concerned about the depletion or the levels of NADH and NAD+. Those levels are different in cytoplasm and mitochondria. So that can change how our enzymes would function. So we need to have different enzymes to do the same job, but under different circumstances. And one clear example that we saw about this in the regulation of glycolysis was comparing hexokinase and glucokinase, or specifically hexokinase 1 and hexokinase 4. But when I told you about glucokinase and hexokinase, I felt like I was kind of giving you an incomplete picture. And it's not so much that we weren't going to talk about it, it's just we really start talking, we really get the why for that's the case at the end of this talk, at the end of this set of slides. So I wanted to take a quick minute and talk about why we need something different in the liver than what we need in the muscles. And it has to do with a different role that the liver and the muscles play in the body in terms of glucose metabolism. So it turns out that muscles are really needy. So when do we use our muscles? We use them when we exercise. God knows I use them in bar class on Saturday. Maybe when you're working out, when you're walking, you're trying to catch the shuttle. So muscles are constantly needing to churn through glucose to literally keep our bodies moving. The brain is even needier because whereas some muscles can have a little more variety in how they get their energy, the brain can only do glycolysis, so it can only use glucose. So it needs glucose as an energy source, and the brain actually needs to get glucose from elsewhere in the body, and sometimes even the muscles are going to need glucose from elsewhere in the body, and this is the job that falls on the liver. So the liver needs to respond to the levels of blood glucose, that are the levels that the blood glucose. So it needs to make sure that concentration stays where it should, but it also needs to make sure that glucose gets to the other parts of the body that needs it. This becomes especially important because if you remember, glycolysis is done all over the place, but gluconeogenesis is only performed in the liver and in the renal cortex. So when our body, when we're fasting, like right now, I feel like I'm fasting even though I just had breakfast. I don't know why. If my blood sugar gets low, it's been a while since I've eaten, maybe I'm really busy and I forget to eat lunch, my liver is going to start doing gluconeogenesis, so that glucose will get sent to other parts of the body. And so that's the one thing that the liver does that the rest of the body doesn't do, except the kidneys. But it's also a major source for glycogen storage. So whereas the muscles are like, glucose, break it down, or slow down, stop breaking down that glucose, let's wait until we run out of more ATP to do our job, we're fine, we're resting. The liver has to keep track of breaking down glucose, building it from nothing, or building it from pyruvate or other downstream products. And it also has to think about storing glucose as glycogen or breaking down glycogen to make more glucose. And so that's why we're spending a lot of time thinking about the liver now. And hexokinase is a really important control point because hexokinase is the step that catalyzes, that turns glucose into glucose 6-phosphate. And it turns out that glucose 6-phosphate, once it's glucose 6-phosphate, it can go through glycolysis, or it can get turned into glucose 1-phosphate and then be turned into glycogen. So this junction where you go from glucose to G6P is really important for determining the fate of glucose in the cell, which is one of the reasons why the liver has a different isozyme than the muscles, because we have to have an extra layer of control. Because if we think back, okay, I'm going to go back to another PowerPoint. I think I'm going to go back to another not Camtasia. Okay, did we talk about it in metabolic regulation? No, we didn't. I'm going to have to go back even further. Gluconeogenesis. Go back here. Okay, we may not have gotten there yet. Did we talk about glucose 6-phosphate and how it gets transported in the liver to make sure glucose gets into the bloodstream? Have we gotten there yet? Okay. Does anybody remember where that figure is? I am drawing a blank. Sequestration. I think it's in gluconeogenesis. What am I doing on time? Oop, I'm eating up time. Eating up time. Okay, we will find it. But either way, the liver, so when this glucose gets turned into glucose 6-phosphate, it's going to get shunted into either of these places. But the liver has to make sure it has a special mechanism for making sure that glucose gets turned into glucose 6-phosphate in a special part of the liver where it won't get turned into glucose and go through or it won't get shuttled through glycolysis, but rather that it will get sent out into the cell as glucose. So the liver, there are lots of controls to make sure that glucose can get out of the liver when it needs to, not just be sent through glycolysis to make pyruvate that the liver doesn't need. Does that make sense? Does anybody have any questions about why the liver is so complicated? We can also, if you have some confusions, we can also put them on hold and keep going and then bring them up at the end of class. But I just wanted to provide a context for why this would be the case. And we'll see this again. We're going to circle back. It'll get hammered back in. Okay. All right. No, you're thinking about the exam? Okay. Totally get it. Okay. We will go back to my little PowerPoint, and we're going to talk about hexokinase and glucokinase one more time. So again, if we can remember, G6P, glucose 6-phosphate, can either go into glycolysis or it can be put into glycogen. They also make sure that it can be turned into glucose that can get sent out into the bloodstream. So once it turns into G6P, it doesn't just get sent through glycolysis. And so hexokinase is used for glycolysis in muscles. Glucokinase turns glucose into glucose 6-phosphate. That can get shuttled through glycolysis, but it also can be put into glycogen in the liver. Because then again, we talked about how the liver does all kinds of crazy stuff. And so because of these differences in their jobs, hexokinase and glucokinase are going to be different in the way that they respond to glucose levels. So if you're in the muscle and you have glucose, remember the muscle can't do gluconeogenesis. They have some glycogen, but not quite to the extent as the liver. So when hexokinase in the muscle sees glucose, it's going to be like, bam, let's turn that glucose into pyruvate. We need to do some movement. And so it has the Km that's really, really low. It's way below the level of glucose in the cytosol. So once it's already going to be operating at Vmax. So when it sees glucose, it's going to go bam. You think of that being like bam. It's going to turn that glucose into pyruvate. one of the ways that hexokinase is regulated is it is actually inhibited by its product, glucose 6-phosphate. So if you're checking through glycolysis in the muscle, hexokinase is running really fast. The way that it gets turned off is if there's too much of a buildup of its product. So if we're doing fine, we have lots of downstream products, we have more glucose 6-phosphate than is being used, we're going to see product inhibition, where glucose 6-phosphate is going to bind to hexokinase, presumably allosterically regulating it and being like, okay, let's turn off, let's take it down, buddy. And then once the glucose 6-phosphate levels go down, the glucose 6-phosphate will pop out, and then glycolysis will proceed as normal. And that makes sense. If hexokinase is not very sensitive to the levels of its substrate, it makes sense that its product would come back and turn it off. And again, because hexokinase is going to break down glucose when it's needed, because that's just its job. Now, glucokinase has to be a little more subtle, because again, the liver is going to have to adapt to things. And keep in mind that these are just two examples. I believe that there is another form of hexokinase in the liver. But for glucokinase, when glucokinase has a really high Km. And so it has a Km that's pretty close to what the concentration of glucose is. So when it sees some glucose, it will turn it into glucose 6-phosphate. But if there's a lot of glucose, it will metabolize it. But if there's not a lot of glucose, so let's say there's a blood, then it's not going to work quite as well or proceed as quickly. Now, if there's low blood glucose or just low glucose in the liver, how is the liver going to respond to that? Does anybody want to venture a guess? It's related to the thing that the liver does that nothing else does. So if we don't have a lot of glucose, what's the liver going to want to do? Is it going to want to make more glucose, or is it going to want to break that glucose down? It's going to want to make the glucose. So we don't want glucokinase to be super active when there's not a lot of glucose, because we want to make glucose. So if the liver is making glucose to then shuttle it out, we don't want hexokinase to just like undo that. So it's not going to be very active yet. And so it's not going to be very active to begin with. But then there's another level of regulation there where you actually have the regulatory protein coming in, binding to hexokinase, and essentially pulling it away from where glucose is being metabolized. So it's another level of preventing hexokinase from undoing the work of gluconeogenesis. So that's why we have this regulation in place. So high glucose prevents it from being sequestered. When there's a lot of glucose, you want it to get turned into glucose 6-phosphate, so it can either go through glycolysis or it can get turned into glycogen. If there's not a lot of glucose, chances are the liver's probably making more glucose. So you don't want that to not happen. So you need to pull it away. You also, if there's not a lot of glucose, you also don't want it being turned into glycogen. So again, the enzyme is just pulled away and like hidden, being put into like the crawl space where it is hidden from where it can do damage. Crazy ant in the attic. Okay, yes. So, take-home messages. Isozymes are two different enzymes that perform the same reaction. They're structurally distinct, though they can be similar, and they're regulated in different ways. They can be regulated in different ways because they have different structures, but also this comes from the fact that they live under different conditions. And if you want to know a fun fact about hexokinase 4/glucokinase, at least according to Wikipedia, which has citations, we talked about when we first talked about protein and enzyme structure, how to be allosterically modulated, you have to have a multi-mer structure. Glucokinase is even monomer. So that's an exception to the rule. Which you don't have to know that, that's just a fun fact. Because science wouldn't be science if we didn't have an exception to every rule we put out there. So it seems. Okay. So now that we've done that deep dive on isozymes and glucokinase versus hexokinase, and I think I've convinced you about how different conditions can lead to different functions and therefore different regulations, we're going to jump back into where we left off. And where I believe that we left off was, is that right? Okay. All right. So we are talking about how if we look at glycolysis and gluconeogenesis, this step here, step number 3 in glycolysis, step number, oh goodness, we're not going to count. The opposite step is the most regulated because it is the point of no return for glycolysis. Once fructose 6-phosphate gets turned into fructose 1,6-phosphate, there's no going back. Glycolysis is happening. Okay? So that's why we're going to see it has like 3 levels of regulation directly at that spot. 3, possibly even more. There's the most levels of regulation there. And so we went through and we talked about how all the different things and different factors that can cause the reaction to be turned on or off. We talked about the role that ATP and AMP play. Then we talked about how citrate, a citric acid cycle intermediate, can come back and inhibit it because, again, it says stop, we don't need more ATP, slow down, buddy. But we also introduced this kind of seemingly random molecule, fructose 2,6-bisphosphate. It's not an intermediate in glycolysis, but it is a molecule that comes from a metabolite that comes back and then adds another layer of regulation to this step. We were on how is fructose 2,6-bisphosphate made? Okay, so we have fructose 6-phosphate. So it turns out fructose 6-phosphate can be like loaded away in the enzyme PFK2. If you're again, this can be remembered that PFK2 is involved in turning on PFK1 because it uses ATP to phosphorylate fructose 6-phosphate to make fructose 2,6-bisphosphate. So we could think of this step as upregulating P, F, K. I'm going to put all of the dashes in the wrong place, 1. Because it's going to make fructose 2,6-bisphosphate, and fructose 2,6-bisphosphate activates PFK1, which then activates glycolysis. And I'm going to go back and I'm going to keep writing this in over and over and over again so we can get practice because, again, there's a lot of regulation, and we're just going to keep building more levels onto this regulation. Now, fructose 2,6-bisphosphate can be turned back into fructose 6-phosphate by FBPase 2. So FBPase 2, again, it's not an accident. Looks like FBPase 1. So FBPase 2 will remove a phosphate, turn it back into fructose 6-bisphosphate. That is by taking away fructose 2,6-bisphosphate. This will be activating FBPase 1, which should be favoring gluconeogenesis. Now, so if these two enzymes are working in opposition from each other, they're working on opposite pathways, how can they regulate to turn these enzymes on and off? The answer is not easily accessible on your cell phone. So, turns out that the answer would be through phosphorylation, all right? So, again, I know this is dry. I'm going to do my best to be excited about it because it is really interesting, but we just got to do it and we got to get through it. So, it turns out that the answer to this is phosphorylation. So, again, if we remember this enzyme, PFK2/FBPase2, It's actually one enzyme. It just has a split personality. Half of it is PFK2, half of it is FBPase2. They cannot be active at the same time. So adding a phosphate is the switch that turns one site on and the other side off, and vice versa. So it turns out that the inactive form, that the dephosphorylated form, so the no phosphate, okay, the PFK2 is activated. So, and it turns out that the phosphorylation, so the act that activates this phosphoprotein phosphatase, that signal for the phosphoprotein phosphatase to remove the phosphate to turn on PFK2 comes from insulin. So insulin is released when we eat a lot of sugar. There's going to be a sugar rush to eat food. There's going to be a sugar rush to our body that's going to want to trigger glycolysis to make it going. So one thing that you can think about to help keep these straight, because there's a lot of layers, I always like to say, think of the beginning and the end goal, or even just the beginning and the intermediate. So if you can tie insulin to like PFK2 being active, it can help you connect parts of the pathway. So insulin turns on phosphoprotein phosphatase, so PPP. That's going to activate PFK2, which is going to increase the levels of will increase the levels of F26BP. That in turn will upregulate PFK1, which will increase the level of glycolysis. So, the release of insulin is going to promote glycolysis. It's also going to inhibit gluconeogenesis. Yes, great. Okay, so if we go back, okay, so fructose 2,6-bisphosphate is a modulator that activates the activity of PFK1. Does that make sense? Does that answer your question? And it also inhibits FBPase1. So it's turning on glycolysis and turning off gluconeogenesis by inhibiting the enzymes, or by activating and inhibiting the enzymes at that step. I'm sorry, I'm doing a lot of wax on, wax off today. I don't know why. Okay, so that's how the body would respond to an insulin signal. Now, is anybody here familiar with glucagon? I'm feeling particularly familiar with glucagon right now. Glucagon is the signal that indicates that you have low blood sugar. Okay? So, what glucagon is going to do, so if we have an increase in glucagon is going to take the PFK2 FBPase2 and add a phosphate onto it, which is going to activate FBPase2. Now, if we activate FBPase2, what's going to happen to the concentration of fructose 2,6-bisphosphate? Is it going to increase or decrease? It's going to decrease, okay? All right, so we're going to decrease the F26BP concentration. Okay, if F26BP goes down, what's going to happen to FBPase1 and PFK1? Let's think about FBPase1. If we remove F26BP, what does F26BP do to FBPase1? So if we're taking away the thing that is inhibiting FBPase1, what's going to happen to the activity of FBPase1? It's going to increase. So we're going to be increasing the activity of FBPase1. And if we inhibit the activity If FBP is 1, which pathway is glycolysis or gluconeogenesis? The answer is on the slide. Gluconeogenesis. Okay. So these are the levels of regulation that are influencing this. And it's because this step is so important, because once you pass through this step, there's no going back the other way through glycolysis. So that's why we literally have, okay, 1, 2, 3 layers. But you could draw it so it could be more, but there's literally like 3 layers of regulation going on here, whereas the other ones are perhaps a little less convoluted. It's all downhill from here, I assure you. But these are all the levels. So I've got it on tape. Practice going through the slide, thinking through, figure out a way to graph it out or make a network or a web. I have some pictures at the end that show it in like a tree format that might help you. But this is the level of logic that you should be thinking about when you're thinking about the regulation. And for me, it always helps to tie glucagon, like tying the signal to an intermediate step to the end product. That usually helps me keep everything straight. And they make it easy because because 2 of the enzymes have the same name that do the same thing, or not do the same thing, but that activate. Okay. All right. Do we have any questions? Because we're going to move on to something different now. We're going to move on to a different aspect of regulation. Good, move on? Okay. All right, so the last step of glycolysis is pyruvate kinase. So pyruvate kinase, so I'm going to start with how pyruvate kinase is regulated in all tissues first on the right-hand side. So again, pyruvate kinase is the last step. We turn PEP into pyruvate, and so it turns out that this step exhibits it would be feed forward activation. So feed forward activation. F16BP, the product of that committed step, goes and turns on the pyruvate kinase. It says, whoa, buddy, we're going to have a lot of PEP coming soon. You need to up your game, dude. Okay, you need to become more active and get ready to break down these glucose byproducts, these glucose metabolites. Okay, so that's an example of feed-forward activation. But we also have examples of feedback inhibition. So it turns out that the downstream products of this reaction can come back and inhibit it. And it's basically the body saying, ATP, intermediates. We don't need you to process more PEP, slow it down. And so this step is inhibited by acetyl-CoA and also long-chain fatty acids and ATP. Again, these are all further products of pyruvate. And then also, fun fact, pyruvate through transamination can be turned into alanine. So alanine also can come back and inhibit this step as well. So whereas we saw this complicated thing with lots of levels of regulation and hormones and whatnot, pyruvate kinase is regulated by the concentrations of previous metabolites, but also downstream products. So it's a different mode, and it makes sense that this step would be regulated by the downstream products and substrates, whereas the committed step would have to have like extra levels of regulation here. Now, we remember the muscles consume glucose. The liver has to think about sending glucose elsewhere, preserving that glucose, making it, and then sending it out. So the liver has an extra mechanism that it uses to regulate this step. And it turns out, unsurprisingly, is triggered by glucagon. So again, the liver, there's low blood sugar. Glucagon is like, hey, liver, we don't have a lot of blood sugar. You need to make some glucose through gluconeogenesis. What's going to happen is glucagon is going to activate pKa. pKa sound familiar to anyone? Protein kinase A. It's involved in G protein signaling. Turns out glucagon binds to a GPCR, which eventually activates pKa. And if you remember, pKa phosphorylates the crap out of everything. One of the things that pKa phosphorylates is pyruvate kinase. So pyruvate kinase will get phosphorylated in response to glucagon. It'll get shut off, preserving, so essentially turning off the step that opposes gluconeogenesis and allowing PEP to go backwards, if that makes sense. It prevents it from going through that last irreversible step, and instead it can be shifted in the opposite direction in gluconeogenesis. Does that make sense? Anybody else? I know it's a lot. It turns out that molecules that are the downstream state of pyruvate can also regulate where pyruvate goes. So ultimately, its allosteric regulation is going to determine which way pyruvate is going to go. So pyruvate can eventually be made into fatty acids. So if you have acetyl-CoA, which is involved in fatty acid synthesis, it can come back and say, hey, pyruvate dehydrogenase complex, which is turning pyruvate into acetyl-CoA, stop. We have plenty downstream products. We don't need you to keep going. Take a break for a while. Let the body catch up. Acetyl-CoA can also go and it can stimulate gluconeogenesis. So again, it can say, okay, we're fine. Why don't you take that pyruvate and make some more glucose? This is another way in which glycolysis can be turned off and gluconeogenesis can be turned on. Because again, we don't want glycolysis and gluconeogenesis to be running at full speed at the same time. They would basically be undoing each other, and it would be a waste of energy. Okay. So we're going to shift gears a little bit now again, and we're going to start thinking about how we can store glucose long term. I'm going to say let's take a 3-minute stretch break, and then we can get back to business on talking about glycogen storage. So go get some water, go to the bathroom, take a stretch, text your mom, whatever you. Okay. Does anybody have any questions while I'm standing up here? Oh, no, I don't want to play the music. That's the problem with smartwatches. They can do so many things. I actually got a notification that a family member died. Yeah, I couldn't do that. I had to keep track of time. But I'm done that. Okay, Elena, I'm gonna ask you. What is the meaning behind the shirt? So oh right, that was so like yeah, really trying to describe it. Time in line? No. I saw one person for Christmas. I mean, they do, but like they don't. Like they got picked for that reason. That's fairly cerebral. I like it. I like it. Oh yeah, just a quick reminder. So I know we're coming up. So we've got our exam coming up, and then finals are going to be sneaking up on us to ambush us out of the bushes. But so you guys, I know some people like to study at Wake downtown, and I think the classrooms are like Amanda in the front office will be putting up signs to know when classrooms are available to study. So study and such, you guys are more than welcome. But one thing I will say is if you use the whiteboard, just make sure you wipe them down after you're done using them. So the cleaning staff will clear off the whiteboards at the end of the day, but nobody comes in the morning. So if you're just hanging out here, and I've come in and I've seen some scribbles, they might be from other classes or other professors, but friendly reminder, and I'm going to try to start doing that to make sure you clear the whiteboard when you're done for the next person who uses it. So just a friendly service, a friendly PSA there to think of the person after you. Yes, yes, Alex. Yes, so that is considered, that was prep work for the discussion. Yes. Yeah, yeah, no. I mean, that's more, that was for the discussion, and also that was more like an FYI, so you know about it. It might also help you when you're reading the book. Because that's one of the things that's interesting about the book is he does a really good job of explaining what they do for a non-science audience. Yeah. But sometimes we may get a little confusing, so if you can help. That's one thing. And you can back because some of it, yeah, it got confusing. Maybe they're just trying to infer too much. Oh yes, if you have quiz directions, you can turn them in on the ancillary side table over there. Okay. Oh, okay. I don't know a lot about hockey, so I would explain why I didn't know what they are. I recognize Carolina Hurricanes and like Red Wings. That's about it. Cleveland had a hockey team when I was a kid. I can't remember what it was. What row? Okay, I think that's been a 5 minute break. Has it been 5 minutes? I think it's been about 5 minutes. Yeah. Okay. Right? I'm sure. I was like, I feel like when I looked at my watch, it was 9:53 and it's 9:58 now. So I'm gonna go ahead and I'm gonna get started, and the information is on the slides. Okay, so glycogen. Glycogen is how our body takes glucose and shuttles it away and hides it for use on a rainy day. So we talked about how the liver can do gluconeogenesis when it's like, oh no, there's no glucose, let's make more glucose. But gluconeogenesis, as we see, is fairly costly. Fortunately, gluconeogenesis is not the only way that our body can get more glucose to break it down and do glycolysis. Turns out that our body can also access glycogen. So it can take glycogen, and it can break it down, and then use the glycogen. And so we talked about glycogen vaguely. We know it's there. We know it's a resource. Today we're going to know what glycogen looks like, and we're going to talk about how glycogen can get added onto and then broken down into individual glucose units. And again, when we're talking about glycogen here, all the stuff with glucokinase might start to make a little bit more sense. Okay, so glycogen. We're talking about glycogen. We're talking about how glycogen is upregulated and downregulated as need or broken or built onto, depending on the situation in the body. We're going to be thinking about the breakdown reaction, which is catalyzed by glycogen phosphorylase. The enzyme responsible for making, I'm just going to put here, is glycogen synthase. And we're going to talk about glycogen synthase too in detail as well. but we're going to start with talking about glycogen phosphorylase. And so if we have glycogen, remember glycogen is this spiraling thing that has lots of free ends, and the free ends in glycogen can be snipped by glycogen phosphorylase to have individual sugars that can then be sent through glycolysis. So the product of this reaction, though, when you break down glycogen, is glucose 1-phosphate. Now, is glucose 1-phosphate an intermediate in glycolysis? No. Glucose 1-phosphate is not an intermediate in glycolysis. So glucose 1-phosphate has to go into an extra step. It doesn't get turned into glucose. The phosphate gets moved from the 1 to the 6. It just takes a little hop and moves up to the down to the 6. And then once glucose 6-phosphate is produced, it can go two ways. It can go into glycolysis, or it can be dephosphorylated by glucose 6-phosphatase to go into the bloodstream, and this happens in the liver. So again, we remembered it's not going to get turned back into glycogen because it's already been taken from glycogen. So it can either go into glycolysis in the muscle or the liver, and then the special option in the liver is to be dephosphorylated and then shunted out. So what exactly does glycogen phosphorylase do? Glycogen phosphorylase being the enzyme that chops up glycogen into glucose 6-phosphate, which goes to glucose 1-phosphate, then goes other way. Glucose 1-phosphate gets turned into glucose 6-phosphate, goes through glycolysis to transport it out to the bloodstream. Well, what glycogen phosphorylase does is it finds one of the many ends on glycogen. It snips that bond by putting a phosphate onto the anomeric carbon of the last glucose, and then a new non-reducing end on glycogen is produced. So glucose 1-phosphate, and again, this enzyme is putting a phosphate onto that last glucose. So this enzyme would be an example of a transferase. But glycogen is a complicated molecule. We remember it looks something like this, and then it has branches. So there has to be a way that the enzyme branches. And so what happens is when you snip off the glucose 1-phosphates, they get turned into glucose 6-phosphate by phosphoglucomutase. And then they get put through glycolysis in the muscles, which goes for energy. And so what'll happen is glycogen phosphorylase will just keep snipping down a branch of glycogen until there are 4 glucose monomers left on the chain. Then you have to have some other enzymes come in and take care of the chain. So you have the debranching enzyme has a transferase activity. And what it'll do is it'll snip between glucose 1 and 2. Those 3 yellow glucoses will get transferred to the end of a new branch. So the purple branch now has 3 more glucoses tacked on the end. But then you still have this lone glucose branch guy. And so the debranching enzyme also has a glucosidase activity, and what it'll do is it'll go in and then snip that. It'll snip that linkage, the 1,6 linkage, and that will be released as glucose. And then you have a new branch or a new string that doesn't have a branch, and you'll have glycogen phosphorylase come in and snip, 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 snip until it reaches the end again. And then the process will repeat itself until eventually you run out of glycogen. Okay, and this is the slide that I thought we talked about already, but we haven't talked about yet. And this is one of the reasons why glucose, because again, we talked about how the liver needs to take glucose and put it into the bloodstream so it can go to the brain and the muscles. Now, glycolysis happens in the cytoplasm of the liver. So if we have glucose, it's turned into glucose 6-phosphate. Now, we don't want to put glucose 6-phosphate into the bloodstream. We want to put glucose. Now, what would happen if glucose 6-phosphate was dephosphorylated in the bloodstream or in the cytoplasm? If that guy was turned into glucose, does anybody know what would happen? It wouldn't be able to leave, and why wouldn't it be able to? So glucose, so if G6P is turned into glucose, in theory, it could still bind to the transporter. That's okay. That's a good line of thinking. You're thinking about could it. So it's not so much that it couldn't bind to the transporter, but you're heading in the right direction. It's that it couldn't get to the transporter. Because what would happen is hexokinase would come in, or the opposite reaction would come in, and it would turn, if that reaction got turned into glucose, it might get turned back in and undo the reaction and rephosphorylate it. So we have to worry about glucose, the reaction being undone out in the liver, because again, the liver is doing 50 million things at once, and depending on whether or not it needs glycogen, like it might turn it into glycogen, or take it another direction, or just put it through glycolysis, or not glycogen, but it could put it in glycogen, it could put it through glycolysis, different things could happen. So what happens is there's a special transporter between the cytosol of the liver cells and the ER lumen of the liver cells. And so glucose 6-phosphate goes to the glucose 6-phosphate transporter. And then once it's safely in the lumen and isolated from glycolysis, it gets turned into glucose by this membrane-bound protein, glucose 6-phosphatase. Then, once glucose is in the ER lumen, it will go through 2 glucose transporters, one in the lumen of the ER, one that separates that membrane between the lumen, ER, and cytoplasm, and one that separates the cytoplasm from the bloodstream. And it goes, and then it can go to other cells. So this is what the liver is dealing with and why it has to have all these tricks to make sure that the glucose makes it out instead of getting trapped in perpetual glycolysis / gluconeogenesis / glycogen storage. So these are all things that our body is dealing with. And so again, we don't want glucose 6-phosphate to get funneled. So if we were to have the glucose 6-phosphatate out of a cytoplasm, it makes glucose. It would get turned back into glucose 6-phosphate, and it would just repeat itself. So we have to sequester it and the ER lumen. And ultimately, we need this because the liver is responsible for keeping, either raising or keeping, for keeping a relatively constant level of glucose in our blood. Okay. So I told you that we were going to move into glycogen. And so this is the general pathway for how we make glycogen from glucose. I need to update this figure because it's blurry. So we know, so the first step of glucose going into glycogen is being turned into glucose 6-phosphate by glucokinase. So now we don't have glucose, we have glucose 6-phosphate. Again, so then once we have glucose 6-phosphate, phosphoglucomutase turns it into glucose 1-phosphate. Now, once we have glucose 1-phosphate, we've taken that glucose out of the pool of glucose that funneled glycolysis. Because in theory, it's reversible, it could go back the other way, but now we're at a point where we're probably going to be turned into glycogen. So the way that glucose polymers are made is that the way that glucoses are linked together to make glycogen is you take uridine triphosphate and you attach the sugar to the nucleotide. And then the nucleotide becomes the leaving group when you want to link the 2 sugars together. And so eventually, so the sugar nucleotide will be added to a growing branch of glycogen, and eventually you'll get long branches, and they cut off after a certain point. I can't remember what the magic number is, but it reaches a certain point where you've reached the maximum length of the glycogen branch, and then it stops and it moves on to another branch. And so ultimately, if any of these enzymes have a weird mutation that has them from working properly, you can have glycogen storage disease. So these are very important. And so again, with here, we're making glucose, where we're adding the UTP and we're making UDP glucose. We're activating the glucose monomers so they can be added together. Just a quick note on Carl and Gertie Corey. They were a husband-wife team, and they were the scientists that really did the seminal work on discovering this pathway. They won a Nobel, and then actually trained 6 scientists who would go on to get Nobel. So basically, they were badasses. Okay, so what does the formation of a sugar nucleotide look like? So if we go and we label our phosphates, alpha, beta, gamma. Now, because again, alpha is closest to the nucleobase. Now, we want to attack, we want the sugar. The sugar already has a phosphate on it, and we want to attach the sugar to a nucleobase. So if we want to attach the nucleobase, we're going to attack the phosphate that is the closest to the nucleobase. So the sugar phosphate is going to make a nucleophilic attack onto that alpha phosphate, and then the beta and gamma phosphates say bye-bye, and they leave as inorganic pyrophosphate. That eventually will get broken down to make that. But now we're left with a sugar nucleotide, or an NDP sugar, where N is a nucleotide and DP comes from the diphosphate. But now that the sugar has a nucleotide diphosphate attached to it, it is ready to be added to a glycogen chain. Oh man, here's the big deal. And so this is where glycogen synthase comes in. So what glycogen synthase does is glycogen synthase takes the UDP glucose and adds it onto the non-reducing end of a glycogen chain. So there has to be more than 4 residues on that chain to have it added in this way. And so if we see this hydroxyl, and then that carbon number 4 and carbon number 1 are going to be attached to make that glycosidic linkage there. And we're not going to go over the mechanism for this particular one. But what we are going to talk about, so we know glycogen synthase is responsible for tacking on the next glucose. What we are going to talk about, though, is how exactly does glycogen get started. So it turns out that glycogen is not just made of carbohydrates. Glycogen is actually a glycogen, or actually a carbohydrate sugar combination. And so in the center, if we draw glycogen, we draw it like with a knob in the center, and then with these little tendrils coming off, and they're branched. Turns out at the center of glycogen is a protein called glycogen. And so glycogen is responsible for nucleating the formation of glycogen polymers, and it acts as both the primer, so it provides a template, and it also acts as the catalyst for the reaction. So what happens is glycogen has a tyrosine 194, and so tyrosine 194, conveniently, it's flat and has 6 rings like glucose. So the hydroxyl group on the tyrosine is nucleophilic. So it's going to make a nucleophilic attack on that carbon of UDP glucose. And so the uracil, or the UDP, is going to leave. And then you have glycogen, which has a glucose attached at that tyrosine hydroxyl. So now that first glucose is attached at the tyrosine, it has a hydroxyl group free that can then go and make a nucleophilic attack on the next glucose. So it makes a nucleophilic attack on the next glucose, the UDP leaves, and then it repeats this 6 times to get glycogen started. So it's being the primer, it's the template for the reaction, but it also gets it started by performing that first nucleophilic attack that then enables the next glucoses in the chain to make the next nucleophilic attack. So this is just a diagram of what glycogen looks like. So in the center, we have you have one glycogenin per glycogen unit. So you can see here we have glycogenin in the center. That first chain, I think, is 8 approximately glucoses long, and that's the primer. And then off of that first strand, you have branches. It looks like each has 2 branches that you can branch off, and then you have layers and layers of glycogen chains. Each of the glycogen chains are approximately 12 to 14 glucoses long beyond that first primer strand. And then you can see here the body can come in. There's lots of non-reducing ends that are available to be snipped. And in a time of need, the body can come in and snip away at that using glycogen phosphorylase. And then there's lots of sugar hanging around, the body can then add back using glycogen synthase. So it's got a lot of ends to pull from. Now, so glycogen breakdown and synthesis are regulated in opposite ways. And the targets for this are glycogen phosphorylase, which breaks down the glycogen, and glycogen synthase, which makes the glycogen. So there are 2 levels of control. Thinking big, we have the global control. So we can have hormonal signaling, being like, dude, I need more glucose, let's break down some glycogen, or we have too much glucose, let's make more glycogen. Or you can have phosphorylation controlling enzyme activity. And then some local controls are that the enzymes are modulated by precursors and products, or they could be allosterically modulated as well. So if we think about the pathway to regulate glycogen phosphorylase, so we're going from glycogen to glucose. The hormones that are going to activate glycogen phosphorylase, if we are wanting to break down glycogen, hormones that are going to say, let's break down some glycogen, are going to be epinephrine, which is the fight or flight. Oh my gosh, I'm scared as a curse word. I need more glycogen to go and fight a bear. Or glucagon. I'm really hungry. I need more glucose to make my ATP. Glucagon is going to say, hey, let's break down, let's make more glucose. But then there's also local control of phosphorylation and the actual concentration of glucose. So, and if we take a look at what the effects of each signal would be, so phosphorylase is active when it's phosphorylated. So we can draw a little, there. And it's not activated when it's dephosphorylated. So glucagon and epinephrine are going to turn on phosphorylase B kinase. That's going to activate phosphorylase A. That's going to increase glycogen breakdown. However, phosphorylase B is less active, and so this enzyme here, phosphorylase A phosphatase, or PP1, is going to downregulate phosphorylase A to make phosphorylase B, which is the less active form, which is going to not favor glycogen breakdown. So covalent modification with a phosphate is going to be the switch that turns this on and off. And ultimately, this is going to be controlled by the signals that say, hey, it's going to be turned on by signals in the body saying, hey, we need more blood glucose. Let's work on this. Now, glucagon is associated with the liver generally, and epinephrine is associated with the muscles. So this is the liver. and this is the muscles. And the signals for both are fairly similar, except they start with a different trigger. And then in the liver, the glucose will get turned into blood glucose, whereas and be sent elsewhere, whereas the muscles are going to use that glucose and shunt it into glycolysis. So again, these signals will increase pKa. It will uprate phosphorylase kinase, which will turn on glycogen phosphorylase, which will increase glycogen breakdown, with the ultimate goal of increasing blood glucose for transport elsewhere so it can go through glycolysis. So again, the liver, I like to think as the parent in the parent organ of the body. It's saintly. It's always looking out for everyone else. We should all be more like the liver, or maybe we shouldn't. I'll leave that up to you. So glycogen, again, glycogen phosphorylase, it hangs out in our liver, but it also functions, it's also regulated by the concentration of glucose. So it turns out, so if we have a lot of glucose, we don't need to break down glycogen. We're fine. We don't need to worry about sending more glucose into the bloodstream because we already have plenty around. So what's going to happen is, I like this drawing because it shows that when the glycogen phosphorylase is activated and it's phosphorylated, phosphates are like curled up inside it. But when there's a lot of glucose, the glucose binds to allosteric sites. It causes a conformational change that makes the phosphates exposed and basically allows them to be clipped off and then deactivates the enzyme. So if we have a lot of glucose, so that's the missing part on this thing right here, is that if you have high glucose, it's going to cause a conformational change in glycogen phosphorylase that will cause the phosphates to be snipped off and deactivate the enzyme. Again, the body's really cool, the body's really efficient, and the point of this slide is just to show that the body can adapt really quickly within a matter of minutes. So if I'm really hungry, I'm going to go into the office, my blood sugar is going to spike, and so my body is going to shift from, oh no, let's break down glycogen, to, oh no, we have to process all that extra sugar that Annalise just ate, and we have to turn it into glycogen. Okay, maybe not for a piece of sugar, but if I went and gorged myself on chocolate, it's probably going to take it and put it into glycogen. So the glycogen breaking down will be downregulated, and the glycogen building up will be upregulated. Glycogen synthesis, turns out we're almost done. Glycogen synthase is also controlled by hormonal signaling, by phosphorylation, and by concentrations of its substrates and products and other related metabolites. So glycogen synthase is also regulated by epinephrine and glucagon, but it has another layer, and that is also affected by insulin. So epinephrine and glucagon, those are we need more. I like to think of it as I'm hungry, but that's not true for epinephrine necessarily. These are our signals that are like, need more glucose. Need glucose. Make an angry noise. And so these are going to, if we want more glucose, these are going to they're going to turn off glycogen synthase. Insulin is like, we have glucose, we need to use it. And so one of the byproducts of that is going to be taking glycogen synthase and making more glycogen. But there's also the same local controls of phosphorylation and substrate products, etc. So what this looks like is, if we have glycogen, so the way whether or not phosphorylation turns on or off is conveniently the opposite of what we saw for glycogen phosphorylase. Glycogen phosphorylase is active when there's no phosphate. So what insulin does is insulin is like put away that glucose, make it into glycogen. So what it's going to do is it is going to prevent glycogen synthase from being turned off. So it inhibits the enzyme that dephosphorylates glycogen synthase. Insulin, glucagon and epinephrine are going to turn off the enzyme that phosphorylates and activates glycogen synthase to be active by removing its phosphate. Insulin, it turns out, is also going to help that process. So insulin is going to prevent phosphorylation and activate dephosphorylation, whereas glucagon and epinephrine are just going to prevent dephosphorylation. It turns out that glucose, and again, here's just a reminder that they're regulated, that glycogen synthase and phosphorylase are regulated by phosphorylation in opposite ways. And then finally, glucose and glucose 6-phosphate. You have a lot of glucose and glucose 6-phosphate. It's going to want to. So glucose and glucose 6-phosphate are going to activate the enzyme that dephosphorylates glycogen synthase and then becomes active. And the last content slide here is just showing you the zoom out on how this is related to the insulin receptor. I'm not concerned with you knowing all of those blobs with abbreviations on them. Sorry, they're important, but they're out of the purview of this class. What I want you to know is that the insulin receptor, there's signaling, something gets phosphorylated that eventually, I wrote it down, insulin deactivates the enzyme that phosphorylates and turns glycogen synthase on, which makes glycogen. So the insulin signal is preventing the deactivation of glycogen synthase. So this enzyme here is going to phosphorylate glycogen synthase. Insulin says, nope, I'm going to inactivate you. So glycogen synthase will stay active. I still have trouble reading these diagrams sometimes. It's okay. If you're looking at it and you're like, whew, sometimes I still get a little disoriented by all of the arrows. It's okay. With time and looking and reviewing and drawing diagrams, it will help you. Yes, Paige. Okay, so insulin takes the molecule that phosphorylates glycogen synthase and phosphorylates it, or not doesn't do that. It phosphorylates other molecules that eventually will deactivate the enzyme that phosphorylates glycogen phosphorylase. So insulin is, oh goodness, so insulin inhibits this enzyme by phosphorylating. So that's all that I have in terms of content. There are diagrams where there's summary of regulation on the last couple of slides. They're all to help you compartmentalize the different things. Here I talk about, or there's talking about glycogen breakdown and glycolysis. We already talked about that at the beginning of class. So what I'll say is look through the slides on your own time. See if any of these methods help you. If you have any questions, feel free to ask either next week or during office hours. Put that off till after the exam. I totally understand. But I would definitely recommend sitting with this over the weekend and beginning to draw out some diagrams to help you to keep it straight. Because going through and practicing, thinking about how hormones regulate these steps are going to help you